Thank you, Dr. Waxman. So, as he said, that is the title of my talk, and I am Alex Zosel. So first off, some background. Daniel Rario is also known as the zebrafish, and the zebrafish is very useful in the study of developmental biology, specifically for its relatively high reproductive rates, fast reproduction, it's comparatively to other model organisms, easier care than others, and that its eukaryotic genome is fairly similar to humans. So if there's a research question pertinent to humans, it can be asked and studied through zebrafish, and they have comparatively easy to manipulate DNA. So next is why I2 and I3. So there's this transcription factor which increases the production of proteins during development of muscle and kidney cells during early development. And what I2 and I3 do they are co-regulators. So they come in and do something to the 6-1 to make either transcription go faster or slower. And we would like to determine what they exactly do and why do they come in, what activates them, and what they're doing. So to do that, we use a type of molecule called a morpholino, and we do morpholino injections. So a morpholino is just a organic molecule that looks right here that is very similar to a nucleotide strand. So it looks very similar, and that allows it to hybridize to a messenger RNA. So DNA undergoes transcription, turns into messenger RNA, and then the morpholino can hybridize and stick to that messenger RNA so that when that messenger RNA is going to go through translation, it can't. So before we go to that, also these are specifically splice morpholinos. So when a RNA is first produced, it is immature and it contains these things called exons and introns. And what has to happen before the messenger RNA is mature is that those introns have to be kicked out and those exons have to be spliced together. So what a splice morpholino is supposed to do is that it will bind in a junction between an exon and an intron, and when those introns get kicked out, it carries an exon with it. So if that happened, specifically in IA2, you'd expect from this region, if we study from exon 5 to exon 7, that if we had a morpholino in there that stuck to exon 6, that when it got kicked out, you would get a product of 252 base pairs instead of the normal product. Conversely, in IA3, if you look at a region from exon 1 to exon 3, it is 271 base pairs. If you lose exon 2 through this splice morpholino, then you get a product of 147 base pairs long. So that leads me to the hypothesis that specifically designed splice morpholinos will do one of the two things or both. They'll do that exon skipping I discussed earlier, so it's akin to having a whole paragraph and you lose a whole section of it. The rest of it still makes sense and it has the possibility of conveying something, but doesn't convey the message at the start. So whatever it does, it's not IA2 or IA3 anymore, so it doesn't function. Or you get that premature degradation of messenger RNA. So that messenger RNA is not getting translated, so there's no protein. So to test this, we do two methods. First is polymerase chain reaction, or PCR. So first we need to obtain embryos from the zebrafish. Then those embryos are injected with morpholinos, and then those injected embryos with a cohort of embryos that are not injected to compare to as a control, we need to isolate the RNA from that. So the RNA is trapped inside the cells and needs to be taken out and purified. Once that happens, you can do first strand synthesis. First strand synthesis is taking that RNA, which is relatively unstable compared to DNA, and changing it to make a hybrid RNA-DNA molecule called cDNA. That allows us to study it further without it degrading. Then you take that cDNA and amplify it with specific primers that I mentioned earlier for the sections of IA2 and IA3 we're looking for, just to make hundreds of thousands of copies of that specific section of DNA. And then you run that all on a gel electrophoresis. So how a gel electrophoresis works is you insert the DNA in these lanes on one end, which has a negative anode, and you run electricity through it, and it goes, since DNA is negatively charged, it goes from negative to positive, and this allows the DNA to separate based on size, as it has to flow through these cavities in the gel. So here you see NIC, non-injected control. That band is fairly bright. There's a normal amount of DNA in there, and due to its coloration. If you see the lane above it, which is morpholino injected, that would be just degradation. It's a lot lighter of a band, harder to see. But the one above it has an alternate splicing. So you not only get the normal size band above this example IA2, you get this band below it, which is where all that lost messenger RNA went. It became alternatively spliced to this different product, which again is not useful. So first we need to do a test run to confirm that our primers were targeting the region that we wanted. So this at the top is a DNA ladder. This just allows us to compare by known sizes of DNA to our samples. So as you can see, the IA2 and IA3 are in the correct locations of what they're supposed to be. So here is the result of the IA2 morpholino test. You got the ladder on the bottom and non-injected right there, but when you see that the morpholino, it is a lot lighter, and you can just faintly see there's a second band there. That means that alternate splicing is occurring there, and you're getting that smaller DNA product. In IA3, however, you see the ladder, non-injected, and then there's two concentrations of injected morpholinos. It starts to look like the injected ones are separating, so there's two different sizes of DNA, but this gel needs to be run out longer as they're way too close as it is. But as it looks right now, it suggests that they are being alternatively spliced. So the second method is quantitative polymerase chain reaction, or qPCR. So here you can just start with the cDNA you made earlier and then amplify it with the same primers, but this time you have a probe. So what a probe does is when polymerase attaches to DNA and is moving along, synthesizing a new strand, it hits this molecule called a probe, and that fires off a fluorescent molecule. That fluorescent molecule can be counted by the qPCR machine as one round of replication. So it can give you a starting value of how many regular RNA, which then was turned into DNA in the cells. So here's the results of IA2. You would expect that non-injected, they would have, as it's relative to the reference, it has 1 or 100% of all that DNA. That's good. That means it has what it's supposed to. But when you look at that IA2 2 nanogram, it has 0.06. That means that the morpholinos are working incredibly well, that almost 94% of the IA2 RNA has been removed, as there's only 0.0613 of the reference. So that means that the morpholinos function. They're doing as they're supposed to, and they're doing the splicing plus degradation. So a couple things that need to be redone. The I3 morpholino gel just needs to be run out longer, so it allows those bands to separate to clearly see the alternate splicing. The IA2 qPCR, so they are done in replicates of 3, the tests, and one of them had a slightly higher standard deviation than the rest, so we would just like to repeat that to make sure that was just due to some sort of pipetting error. So as you can see, there was no I3 morpholino qPCR graph. That needs to be done as we ran out of time, but overall, the methods are successful. The way in Dr. O'Brien's lab, how they study the function of IA2 and IA3 in development of tissues, that through morpholino injections, it works. So that means the research can go on. And I'd just like to thank a few people, my research advisor, Dr. O'Brien, the faculty of the Natural Sciences Department at UWS, and I'd like to give extra thanks to everyone that helped me, Kim Kobar, the other research students that I worked with, and Dr. Leong of UMD and College of St. Scholastic Biology Department for any devices they may have loaned us during this research process. Thank you.